welcome to my lesson on defence against disease. I've designed my lessons around the Edexcel iGCSE Biology 9:1 course, but it is also relevant for various other courses. In this lesson, I'm going to cover how the immune system responds to disease with phagocytes and lymphocytes, which come up in the paper 1. The outcomes here that are in bold will only come up on paper 2 of the separate award science. And for these, we will cover vaccinations and blood clotting. First of all, let's look at the pathogens. A pathogen is an organism that causes disease. There are 4 main types of microorganism that cause disease: bacteria, and for an example of this would be salmonella that you get food poisoning from. There could be viruses such as the dreaded COVID-19, the flu, which occurs every year, and I'm sure you've seen the news about Ebola. Fungi cause athlete's foot and ringworm. Athlete's foot is unpleasant. You can get it especially if you allow your trainers to get very sweaty and you haven't worn socks, and so the fungal spores will stay inside your shoes. And ringworm forms on your skin, and it is a fungus on your skin, and it forms little rings, which is why it's called ringworm, but it's not a worm. And then we've got productista such as malaria, which is transmitted by vectors called the Anopheles mosquito. Now we've reminded ourselves of the main pathogens, let's look at what immunity actually is. Immunity is the acquired ability to defend against infection by disease-causing organisms. In other words, how our body fights the infection using our own cells, our own white blood cells. We are surrounded by microorganisms, and to try to reduce the chance of us getting infected, we have 3 main lines of defence. The first line of defence is the most basic form. It consists of physical and chemical barriers, such as the skin, tears which contain enzymes to destroy pathogens, mucus in the nose and the trachea and the reproductive tract, and cilia, which you may have come across with ciliated epithelial cells in your trachea, which waft mucus, which have trapped pathogens in it, up out of the trachea, which we then swallow down into our stomach. And our stomach has acid in it to destroy any pathogens which are ingested with our food or are swallowed down with the mucus. We also have urine flow, which flushes bacteria out the urethra and also out of the bladder. And then we have friendly bacteria, which we're learning more and more about, the gut biome, where these bacteria compete against unfriendly bacteria that you may consume with your food. So blood clotting is also a first line of defence. When we cut ourselves, we lose blood, and also the break in the skin means that the pathogens can enter the body. Therefore, the body must try to block the break in the skin to prevent blood loss and infection, and for this to be effective, it has to be quick. Platelets in the blood carry an enzyme, and we've talked about platelets earlier on in the course. They're only fragments of cells, they're not actual cells in their own right. But they carry an enzyme, and this enzyme is released into the plasma whenever the platelets come across air or damaged cells, such as a cut. The enzyme changes a special protein that's found in your plasma. You don't see it in your plasma because it's actually soluble, and it's called fibrinogen. And when the enzyme works on this fibrinogen, it converts it into an insoluble protein called fibrin. Well, fibrin is sticky, and it forms long threads, and these long threads make this sort of crisscrossing across the cut, creating a mesh which traps some of the red blood cells in it. And this makes a plug inside that cut, and this is a blood clot. And then of course you get this scab that then forms over the top of this, and then underneath that you get fresh skin starts to form. Well, while all of that's going on, phagocytes, which we're going to be talking about very soon, are attracted to this damaged site, because what they're going to go and do is they're going to go and clean up the site and engulf pathogens. And you may see this in pus underneath the skin. So then we've got the second and third line of defence, which make up the immune system. The second line of defence comes if the pathogens get past the physical and chemical defences. It is still non-specific, meaning it fights any pathogens that enter, and it involves white blood cells, such as phagocytes. Then there is a third line of defence, which involves a type of white blood cell called lymphocytes, which are made of two different types of cells, B and T cells. B cells are made in the bone marrow and T cells are in the thymus gland, so their names come from where they mature, B for bone, T for thymus. But what actually triggers the immune response? All cells, including our body cells and pathogens, have proteins embedded in their cell membrane, and these proteins allow us to recognise our own cells as self, and so, unless we have a fault with our immune system, such as an autoimmune disease, we do not attack our own cells. Pathogen proteins are foreign to the body and they are called antigens. These trigger our immune system, so they trigger our white blood cells to fight these foreign antigens. Toxins that enter our body, possibly from bacteria or from snake bites, can also act as antigens as they contain protein. Phagocytes, being the second line of defence, are the first response to the foreign antigens. Phagocytes are easily recognised by their strange-shaped nucleus, known as a lobed nucleus, which means they can easily pass through the blood vessel walls into the surrounding tissue and move towards the antigens and bind to them. The phagocyte's membrane then surrounds the pathogen and engulfs it, forming a phagosome, which is a vacuole containing the pathogen, phago meaning to eat. Enzymes found inside the cell, held in vacuoles called lysosomes, then go and bind onto the phagosomes, release the enzymes into the phagosomes and break down the pathogen in order to destroy it. They may also send out chemical messengers that help nearby lymphocytes to identify the type of antibody needed to neutralise them. As phagocytes act the same way with all pathogens that they encounter, they are known as non-specific. The third line of defence then involves the special white blood cells called lymphocytes. These provide a specific immune response to infectious diseases. As already mentioned, there are two types, T cells and B cells. Looking at T cells first, some of them help B cells, in other words they're called T helper cells, and they help B cells respond. Other ones, called killer T cells, kill body cells infected by viruses. With the helper T cells, B cells make special Y-shaped proteins called antibodies. Each type of antibody matches to only one type of antigen. So this process can take a few days, during which time you may feel ill. The antibodies work in various ways, but the main ways are that the antibodies stick to the antigen, marking it for attack by T cells, or they stick the pathogens together, making it easier for phagocytes to engulf them. Some pathogens produce toxins which make you feel ill. Lymphocytes can also produce antitoxins to neutralise these toxins to stop you from feeling ill. Both the antibodies and the antitoxins are highly specific to the antigen on the pathogen. Therefore, the lymphocytes that produce them are called specific. The response when you first encounter a specific antigen is known as the primary response, or this primary immune response. It takes a few days before the B and T cells divide and start to attack the antigen, and during this time you feel ill. Antibodies numbers are eventually high enough to help destroy the antigen. During this immune response to the foreign antigens, the B and T cells also make memory cells which remain in your bloodstream, and this means that you are said to be immune. The second time you encounter this same antigen is known as the secondary immune response. The memory cells, which were produced during the primary immune response, recognise the antigen that's reinfected you. They multiply and produce larger amounts of antibodies in a shorter length of time. This immune response is much faster and more effective than the primary response. Sometimes this comes up on exams, and it comes up as a graph, which I want to show you now so that I can talk you through it. This shows the antibody concentration versus the time. So let's have a look at it. Here, first of all, we've got this initial exposure to the antigen that you haven't come across before. And your B cells and T cells are trying to recognise what type of antigen it is, and so therefore they can respond to it and multiply. So this antigen that has entered your body will then be bound onto by T and B cells, which then multiply. And the T helper cells help the B cells, and then the B cells start to produce your antibodies against the antigen. Now this takes a little bit of time, and during this time you may feel ill because they're infecting you, but eventually you produce enough antibodies to actually destroy that antigen, and so you start to feel better again. And then there's no point of you having those antibodies anymore, and so they start to get broken down by your body cells, and you don't produce any more because it's just a waste of energy. However, you do leave memory cells behind, so that when you get a secondary exposure to this antigen, you suddenly get this really rapid response because your memory cells recognise it straight away and start multiplying rapidly, and therefore the antibody concentration dramatically increases. And this stops you from feeling ill because you managed to destroy the antigen before it causes you an illness. So, the best immunity does come from having fought off disease. However, in some cases, such as a serious disease that could prove fatal on the first infection, this is not an option. Instead, we have to use vaccination. A vaccination is an artificial immunity when a harmless or an inactive form of the pathogen is introduced into the body, usually through an injection. This stimulates a primary response, but because the antigen has been inactivated, it cannot actually cause a disease. It just introduces the antigen to the T and B cells. The T and B cells then attack this foreign antigen and make memory cells. So if the actual pathogen or antigen enters the body, the memory cells will recognise it and bring about the secondary response, destroying the antigen before you can become ill. So this vaccination has meant that the person has developed immunity without having the disease. Now, the vaccines can be made from various different forms of the pathogen or antigen. First of all, you can end up with a weakened strain of the actual pathogen, for example, measles, and it's weakened so it can't actually multiply and cause you to be ill. You could use a dead or destroyed pathogen, remembering that you can't say dead if it's a virus because viruses aren't living in the first place, so we would say it's a destroyed pathogen. For example, whooping cough. You can actually just take the toxins that are being produced by the bacteria and modify them so they can't actually cause any damage to the body, and that's an example would be tetanus here. You can take a modified form of the cell membrane proteins themselves, the antigens, for example, influenza, the flu. Or nowadays we can actually take a harmless bacterium and we can genetically modify it and basically implant antigens from the pathogen into the bacteria. But because they're on a bacterium which is harmless, they're not actually going to cause you disease. An example of that is hepatitis B. So that is covering all of the learning outcomes that I said at the beginning. But I'd just like to go through and summarise the work that we have done so far. So first of all, I want to remind you of the 3 lines of defence. So we have the first line of defence, which is your chemical and physical barriers, for example, your skin, mucus and cilia, and blood clotting. Remember, if you talk about blood clotting, you must mention platelets and the fact it reduces blood loss and reduces pathogen entry. It doesn't stop it altogether. The second line of defence are your phagocytes carrying out phagocytosis, and they ingest pathogens, phagocyte meaning a cell that eats. And these go around and attack any pathogen or antigen that is around, so they are non-specific. Then you have your lymphocytes, your T and B cells, and the T killer cells, which kill your infected cells. But then you've got your special helper cells, your helper T cells, which help B cells to respond. And actually it's the T helper cells which are attacked by the HIV virus, which is why it's so clever, because it actually manages to destroy your T helper cells so your B cells can't respond. So T helper cells are very, very important in initiating this third line of defence. The B cells release antibodies, which are specific to the pathogen. And remember that we have these memory cells which are produced after this primary immune response, and they sit around in your body waiting until you come across this antigen again. Now, depending on the antigen, depends on how many memory cells are produced and how long they last. And that is why various different vaccines last for different spans of time, some for only 6 months, some for years. And so this primary and secondary immune response was the basis of vaccination. So vaccination stimulates the production of memory cells, which enable future antibody production to the pathogen to occur sooner, faster, and in greater quantity. That's very important to include in your exam answers. When they're talking about the secondary response, they want that there is a greater number of antibodies produced in a shorter time, and so that it is much more effective. Another thing that's very important for your exams are your biological terms, what I call the buzzwords. These are very important to familiarise yourself with, as you should try to include these in your answers in your exams. Try to learn the spelling too. Well, if you've enjoyed this topic, maybe you'd like to do some further work. Maybe you would like to go and find out about what Edward Jenner, who played an important role in developing the smallpox vaccination using cowpox, hence the term vaccination from the Latin word for cow, vacca. And so that's the end of the lesson on defence against disease.